All right, welcome to chapter 3. In this chapter, we're going to cover some staining techniques and using a microscope. So before we start in that, we're going to first look at just how small bacteria are, which is what we're mostly going to be looking at in this class. So looking at the scale here to the left, we have, this is a red blood cell, so eukaryotic cell. And in comparison, here's E. coli. They have here in nanometers. You'll see some things in nanometers. A lot of times you'll see it in micrometers. If you convert it to micrometers, E. coli would be 1.3 by 4 micrometers. You see that's quite a bit smaller than the red blood cell. Here's another bacterial species, Rickettsia, even smaller. And then here's several viruses, which are quite a bit smaller than that. And if you look at this polio virus here, it's much, much smaller than the red blood cell. This video over here is actually a phagocytic cell that's pursuing, chasing, and eventually engulfing this bacteria, which I think is super cool. So here's some more examples. We have protozoa. This is one of the largest microbes that we're going to be looking at. We did talk about protozoa in chapter 1. Yeast is about 7 to 8 micrometers, about the same size as that red blood cell we just looked at. Bacteria can range in size, anywhere from 0.2 to 1 micrometer by 2 to 8 micrometers. It's going to depend on whether it's more circular or more rod shaped, whether it's going to be longer in one dimension. Some of the smallest bacteria include Rickettsia that we just saw, which is about 0.4 micrometers. This is what's responsible for Rocky Mountain spotted fever. Something that's interesting that we see a lot in these smaller microbes or smaller bacteria is that they can be obligate intracellular parasites. So this is like we see with viruses, how they have to be inside of the cell in order to replicate and survive, where we can see that as well in some bacterial species, especially here with Rickettsia. Also chlamydia, same thing. It's about the same size. It's also an obligate intracellular parasite. This is responsible for STDs and conjunctivitis. And Mycoplasma is another very small bacteria. It's responsible for the walking pneumonia that we see in children. And then viruses, of course, much, much smaller, not even on the micrometer scale. We see those in nanometers usually because it's so much smaller. So here's several practice math problems for you guys. I'm going to let you guys practice that on your own. Hopefully you've covered that in other courses, but if you need help with that, meet with me and we'll talk about it and we'll go over this. Okay, so do practice this because I'm going to expect that you know how to do some of these basic conversions. But for our lecture here today, we're going to focus on microscopy. So there's two main types of microscopy. There's light and electron microscopy. We're going to talk very, very briefly about electron microscopy. We're not going to be doing electron microscopy in our teaching lab, in our class, but I do want you to be aware, so we'll touch on that. But we're mostly going to be talking about light microscopy, which, as the name suggests, uses visible light to observe specimens. There's several types of light microscopy. The first three that's named here, we're going to be doing in the lab. Our microscopes will have this capability. So that includes compound light microscopy, which can be bright field. We also have dark field and phase contrast. A couple more, which was very commonly used in microbiology studies. So we will be seeing lots of images that have used fluorescence and confocal microscopy, but we are not going to be doing that in our lab. So looking at the compound light microscope, which is what we are going to be using. A few different terms that I want you guys to know. So magnification is one. So this is the ability of the microscope to enlarge an object, to magnify that object. Now to determine the total magnification that you are looking at, it's going to be in our microscopes and in most compound light microscopes, calculated by objective lens by the ocular lens. So if we look at this microscope here, light is produced by the light bulb. It's going to come up through the specimen. This is the objective lens here. So the objective lens is going to magnify that image. Then the light continues to pass through, arrives at the ocular lens. And the ocular lens here, which ocular for eyes, right? It's like in the eyepiece, is also going to magnify it even more. So that's why we have to multiply the two lenses together, which both magnify the image to get our total magnification. Now in our microscopes in the lab, which is the case for most microscopes, the ocular lens up here has a magnification of 10. So in order to determine the total magnification, you can look at whatever objective lens you are using, multiply that by 10 to give you your total magnification. That's magnification. Moving on to resolution. So resolution is the ability to distinguish between two closely spaced objects. So if we have, for example, a resolution of 0.4 micrometers, that means that anything that is closer than 0.4 micrometers is going to appear as the same object. Anything that's farther apart than 0.4 micrometers, you can distinguish as two different objects. So this contributes to the clarity of the image. Lastly, we have contrast. So contrast is just the ability to see the object in contrast to the background. And this is helped by staining. So this is why we stain a lot of our specimens, to help increase the contrast of that specimen to the background. All right, so let's look through all these different types of light microscopy. First, we're going to start with bright field microscopy. So this, I remember this term because if you think of the background as the field, then you can think it has a bright field or a bright background. So when you're using bright field microscopy, the image is going to look like a dark object in front of a bright background. Okay. This is what you're going to use when you're looking at stained specimens. So if you ever are in the lab, you perform a stain, and you're going to look at that stain under the microscope. You're going to want to use bright field microscopy. Dark field microscopy is just the opposite in appearance. So now the field or the background is going to be dark. So we're going to have a very bright object, which what we see here is this bacteria looks bright on a dark background. And how this is carried out is by when you switch the microscope over to dark field, it adds this opaque disc here. So as the light comes up from the light bulb, it's going to be blocked in the center by this opaque disc. So the only light that actually reaches the specimen is going to be light that's reached it at an angle. All right. So this allows us to examine living or unstained organisms. Anything that's stained is killed. The process of staining bacteria kills it. So unstained organisms are quite often living organisms. We have phase contrast. And so this is going to have, now we're adding the annular diaphragm or a ring-shaped diaphragm. That's going to create direct rays and diffracted rays. So by having both direct and diffracted rays, that's going to allow us to look inside the organism better. So if you want to look inside an organism, say we're looking at a protozoa and you want to look at the organelles inside of it, then I would recommend that you use phase contrast. Those are the types of microscopy that we will be using in the lab. Another one that's commonly used is fluorescence microscopy. And this is going to use UV light. And first, this specimen has been stained with fluorescent dyes, and then those will fluoresce under the UV light in the microscope. And so that's how we're going to be able to see those organisms better. And then confocal microscopy is very similar because we're still going to be using those fluorescent dyes, still using UV light, but confocal microscope is hooked up to a very complex computer program. And that allows us to actually look at all the different layers. So it'll take an image of each layer through the organism, and then put all those images together to form a 3D image. So these look pretty cool. So if you see these like bright, very clear, you can kind of see the dimensions in the image, that's generally going to be confocal microscopy. So if you see that as we go through the book, that's probably confocal. Very common. All right, microscopy, some practical considerations for you to think about. So different types of microscopes have different resolution levels. So that's going to be important when you're considering your specimen size or the size of what it is that you're trying to look at. That's going to contribute to which type of microscopy you want to use. Also, what it is that you're doing is important. If you're looking at a stained cell, or if you want to look at the internal structures, you're going to use different type of microscopy. Also, with the confocal microscope, when you're staining with those fluorescent dyes, you can label different parts of the cell. So let's say you want to look at the DNA of the cell, you would use a fluorescent dye that's going to tag the DNA a certain color. So that's going to label that DNA. So when you look at it with the UV light, you're going to see that DNA better. So depending on what it is that the scientist or you in the lab is trying to do is going to determine what type of microscopy we use. Also, when you're looking at pictures of or images of different microbes, oftentimes you'll see this scale here. Make sure that you use that to kind of keep in mind the size of those organisms. So it was all light microscopy. And we're going to briefly touch on electron microscopy. So electron microscopy instead of using light, now it's going to use a beam of electrons to create an image of the specimen. Where this is useful is looking at much smaller anything. You can look at DNA or viruses, anything that's smaller, because it has the capability to look at a higher magnification, and it also has a higher resolving power. So we can look at some of these smaller details as well. There's two types of electron microscopy: scanning and transmission. So scanning is used to observe the surface or scan the surface of the structure, whereas transmission microscopy is important for transmitting through and into the specimen that we're looking at. So you're going to use transmission electron microscopy to look at the internal structures. So here's two pictures of electron microscopy. Here we can see that we're looking inside the cell, looking at chromosomes here during bacterial division, sorry, not chromosomes, but we're looking at DNA rather here. So this is going to be transmission electron microscopy. Whereas here we're looking at the surface of this leaf. So that's going to be scanning electron microscopy. All right, so then we're going to start to look at staining. So that was looking at microscopes and different types of microscopy. Now we're going to start to look at the staining that we're going to be doing in the lab. So before we even start to stain, There's a couple of different processes. First, you're going to have to perform a smear. So a smear is the procedure for smearing bacterial or any kind of microbe on the slide. Once you have your microbe on the slide, then you're going to have to fix it onto the slide or attach it. And so that can be done in different ways, but the most common way is by using heat. So we performed our smear, we have bacteria on the slide, and then you're going to heat the slide, and that's going to fix it. And that does two things. One is going to attach those microbes onto the slide, which is really important because as you stain it, if you don't fix it, if you don't attach it, it's just going to wash off. Okay. The second thing that fixing does is it's going to kill those microbes, which is important. It helps us be a little bit safer. So before we can understand how stains work, first we need to recall resting membrane potential. Hopefully you learned about resting membrane potential and how that makes the inside of the cell more negative. That's going to be based on different permeabilities of the cell membrane. It's also contributed to negative DNA. So DNA is negatively charged. There's also a lot of larger proteins that are negatively charged inside of the cell. If you don't remember all of that, that's okay. I just do need you to definitely know that the inside of a cell is negatively charged, okay? Why that's important? Because that's going to affect how different stains behave. So we have two different types of stains. We have basic dyes and acidic dyes. Now, basic dyes are positively charged. And so the positive charge of those basic dyes is going to be attracted to the inside of the cell, which is negatively charged, right? Opposite charges attract. So that's what we see here. The inside of the cell is negative. Our positive dye is attracted to that, so it goes inside of the cell. So it's going to stain the cell itself, stain the inside of the cell. Now that's the exact opposite for acidic dyes. Acidic dyes are negatively charged, so their negative charge will be repelled by the negative charge inside the cell. So your acidic dyes are going to stay outside of the cell, and it's actually going to stain the background rather than the cell itself, which can still be important because as long as you're increasing contrast, it's still helpful. So the first type of stain would be a simple stain. And a simple stain is used just to help to visualize the microbe, make it more or easier for us to see. Along with simple stains or many types of stains, not just simple stains, we might use a mordant. So a mordant is something that's going to help to hold the stain to the cell, or it can also be used to coat the specimen to enlarge it. Differential stain, however, is going to tell us something about the organism, which is going to help us to differentiate or distinguish between different bacterial species. Couple of differential stains that we will be doing is the Gram stain and the acid-fast stain. I'm going to talk about both of those. Now, the Gram stain allows us to distinguish between gram-positive and gram-negative cells. We haven't really talked about this. We are going to talk more about this in the next chapter. In chapter 4, and talk about those peptidoglycan layers. But for now, what I want you to know so that you can understand our discussion here on gram staining, just know that gram-positive cells have many more layers of peptidoglycan. So it has a very thick peptidoglycan layer. And gram-negative cells have just a very thin layer of peptidoglycan. That's the big difference between those two. Gram staining is one of the first things that we do in trying to identify a bacterial species because it's two very broad categories of bacteria. So the gram stain procedure, the first two steps is adding crystal violet and then iodine. Iodine in this case is the mordant. So here, the iodine is going to crystallize or form crystals with the crystal violet inside the cells. So one more time, we're going to take our specimen, we're going to add crystal violet, then we're going to add iodine, which is the mordant. That's going to form crystals inside the cell. In a gram-positive cell, so first, the third step is adding alcohol. Okay, in a gram-positive cell, that alcohol is going to dehydrate the peptidoglycan. So remember, gram-positive has a very thick layer of peptidoglycan, lots of different layers. So the alcohol is going to dehydrate that and compact those layers together. So that then the crystal violet and iodine crystals that are inside the cell, they can't be rinsed out. So that's going to keep that crystal violet in the cell. Crystal violet, like the name is, it's purple. Okay, so those cells are now permanently purple because we've locked it in. However, in gram-negative cells, when we add the alcohol, that's going to dissolve the outer membrane. It's going to leave holes in the peptidoglycan so that now when we rinse, the crystal violet will rinse out of the cell, which is going to decolorize anything that's gram-negative. So any gram-negative cells are going to be decolorized, and the crystal violet iodine crystals will wash out. Let's go through that whole process one more time with this picture, might help. So first we have gram-positive, gram-negative cells. We have smeared them onto the slide. We have heat fixed them onto the slide. Now we're ready to stain. First step is adding crystal violet. That turns dark purple. Then we add iodine. That's going to form those crystals inside the cell. Then we add the alcohol. In the gram-positive, with the thick layer of peptidoglycan, remember that's going to make the crystal violet and iodine crystals, they're stuck inside the cell. So when we rinse, they're going to stay purple. However, in the gram-negative cells, we have the thin peptidoglycan layer. So when we add the alcohol, that's going to create holes in the peptidoglycan layer, and then the crystal violet will rinse right out. And so that's going to decolorize these gram-negative cells during this step. Last step is going to be to add a counter stain. And we're going to add safranin, which is a reddish pink color. That pink color is not going to have any effect on the cells that are already purple, but in the clear cells, it's going to stain them pink so that we can see them. So the final result will be gram-positive cells are going to be this purple color, and gram-negatives will be this reddish pink color of the safranin. That's gram stain. Next, we have acid-fast stain. So this is another differential stain, and it's going to be used specifically for any bacteria that have this waxy material called mycolic acid, you need to know that, in their cell walls. So that's going to be in Mycobacterium and Nocardia species. So all of these species have mycolic acid, and because of the mycolic acid, they don't stain very well with the gram stain. So we do this other stain instead, and it's called acid-fast stain. So the acid-fast stain procedure is going to be first to add the carbonyl fusion, which is red. Then you're going to decolorize. If it's acid-fast, it's not going to be decolorized. It's going to stay red. If it's non-acid fast, it's going to decolorize. And then we add the counter stain, which is methylene blue, which will then turn the non-acid fast bacteria blue. Lastly, there's two special stains that we're going to do. So these are going to be used to distinguish or highlight if a bacterial species has one of these structures. So we have a capsule stain is looking for the presence of a capsule, and a spore stain is looking for the presence of an endospore. Here's the capsule stains. A capsule is a gelatinous covering. and it doesn't really accept dyes. So in order to see if a bacterial species has a capsule, you're going to stain the cell with just a simple stain. So you can see that the cells are stained. And then you're also going to add, like India ink is going to stain the background. That's why we have this, like here's an excellent example here. India ink staining the background, and we also have the cell itself is stained with possibly safranin. But the capsule does not stain very well, and so we see just this clear halo surrounding the cell, which is represented by the capsule. So if you see this clear halo surrounding the cell, then you know that that species has a capsule. And the endospore stain, so endospores are resistant dormant structures inside of the cell. Not all species have endospores, only some of them produce endospores, but you can almost think of it as like a very resistant, hardy like seed that the cell will produce so that it can live in harsh environments. We're going to talk more about endospores in the next chapter. So if that part doesn't make sense to you, don't worry about it. We'll come back to that. But what I want you to know is about the endospore stain procedure here. So the first primary stain is malachite green. So it's going to stain the endospores, which we see inside these cells. This is a beautiful stain here. It's staining the endospores green. Then you're going to rinse that and then add safranin. Safranin is going to stain the vegetative cells. Vegetative cells are the just living normal cells. And that's going to stain those pink. So once the endospore stain is performed, if there are endospores there, the endospores will be stained green and the vegetative cells will be stained pink. And that's it. All right, I hope you've enjoyed chapter 3. See you soon for chapter 4.